we are discussing about the structure of the cell. Okay, endoplasmic reticulum, endoplasmic reticulum, endoplasmic reticulum. Suppose if you consider this is a cell and this is a nucleus. From this outer membrane of the nucleus, there is a membranous reticulum extending throughout the cell. So, it is a membranous network, or reticulum means network extending throughout the cell. This is called as a endoplasmic reticulum, endoplasmic reticulum. It gives a support to the cell. So, it is also called as a cytoskeleton. Cytoskeleton. Just to remember this terminology, cytoskeleton. As this endoplasmic reticulum gives support to the entire cell, so it is called as cytoskeleton. It is also useful for intracellular transport. Intracellular transport means the materials can be transported between the nucleus and the cytoplasm through this endoplasmic reticulum. Okay. So, it is involved in the intracellular transport. Just to remember, it gives support to the entire cell, that is why it is called cytoskeleton. It is also useful for transportation of materials between the nucleus and the cytoplasm. So, it involves in the intracellular transport. There are two types of endoplasmic reticulum. It can be broadly classified into two types. One is rough endoplasmic reticulum, and smooth endoplasmic reticulum. Rough endoplasmic reticulum and smooth endoplasmic reticulum. So, what is rough endoplasmic reticulum? Suppose this is endoplasmic reticulum. If ribosomes are present on its surface like this, if ribosomes are present on its surface, then it is called as a rough endoplasmic reticulum. Rough endoplasmic reticulum. So, that means ribosomes are present on its surface, then it is called rough endoplasmic reticulum. So, the function of ribosomes is protein synthesis, protein production. So, the function of rough endoplasmic reticulum also protein synthesis. Okay. It involves in protein synthesis. Protein synthesis. Just it is for your understanding. You remember, rough endoplasmic reticulum means if ribosomes are studied or if ribosomes are present on its surface, then it is called rough endoplasmic reticulum. The function of rough endoplasmic reticulum is protein synthesis. And smooth endoplasmic reticulum is if ribosomes are absent here, it is plain, then it is called smooth endoplasmic reticulum. And the function of smooth endoplasmic reticulum is lipid synthesis and storage. Lipid synthesis and storage. Just you remember this one. So, just I will revise once. Endoplasmic reticulum is a membranous network extending from the outer membrane of the nucleus up to the plasma membrane, that is called as a endoplasmic reticulum. It gives support to the entire cell, so it is called as a cytoskeleton. It also involves in the transportation of materials between the nucleus and cytoplasm, so it involves in the intracellular transport within the cell. There are two types of endoplasmic reticulums, rough and smooth. Rough endoplasmic reticulum means ribosomes are located on its surface, then it is called rough endoplasmic reticulum. The function of ribosomes is protein synthesis, so the function of rough endoplasmic reticulum is also protein synthesis. Then smooth endoplasmic reticulum means here ribosomes are absent, so the endoplasmic reticulum is plain, then it is called as smooth. Okay. The function is lipid synthesis and storage. Just you remember this one. Okay. So, next one is Golgi complex. Golgi complex. So, this Golgi complex contains some structures like tubular structures, cisternae, tubules, vesicles, okay, like this. This structure is called as a Golgi complex. Here in the Golgi complex, the structures called cisternae, tubules and vesicles. See, these are the components of Golgi complex, you can see. So, cisternae, tubules and small vesicles. Okay. And these structures were discovered by a scientist called Camillo Golgi. So, it was named as Golgi complex. Okay. So, the main function of this Golgi complex is secretion. Secretion means it releases some chemicals which involves in the protein synthesis process. So, the main function of Golgi complex is secretion. It releases some chemicals. Which involves in the protein synthesis. And these Golgi complex, they are also called as Baker's bodies. Baker's body. Just to remember this terminology, Baker's bodies. Golgi complex is otherwise called Baker's bodies. The other names of this Golgi complex are lipochondria, and dictyosomes. So, simply you remember Golgi complex, the main function is secretion. It releases some chemicals which are useful for protein synthesis, protein formation. These are also called as Baker's bodies, lipochondria and dictyosomes. And these are the components of Golgi complex. Just to remember this terminology: cisternae, tubules and vesicles. That's enough. Other names of this Golgi complex, that's enough. Okay, just to remember the terminology, that's enough.
Those three are the parts of it.
Parts of the Golgi complex. Function is secretion. Secretion means it releases some chemical substances which involves in the protein synthesis. Okay, don't go in depth of that one, that's enough. So, Baker's bodies, you remember this terminology. Okay. So, Baker's bodies, these are also called as Baker's bodies, lipochondria and dictyosomes. These are the other names. Right. Next, ribosomes. Ribosomes. So, these were discovered by a scientist called Pallade. So, these are also called as Pallade particles. The main function is protein synthesis. The main function of these ribosomes is protein synthesis. So, they are called as protein factories. Ribosomes, they were discovered by Pallade, so they are called Pallade particles. The main function of ribosomes is protein synthesis, so they are called protein factories. There are two types of ribosomes. One is 70S ribosomes, and 80S ribosomes. So, simply you remember 70 and 80 represents the molecular weight of the ribosomes. S is unit, the name of the scientist Swedberg unit. Okay, just to remember that one, you don't need. 70S, 80S type of ribosomes are present, and the 70S type of ribosomes are present in prokaryotes, that is in the bacteria, and they are also present in mitochondria and in chloroplast. I already told you in the last class. Okay. So, the 70S type of ribosomes are present in prokaryotes, that is in the bacteria, and also in mitochondria and chloroplast of the eukaryotic organisms. And then 80S type of ribosomes, they are present in eukaryotes. They are present in eukaryotes. So, actually these ribosomes are having two subunits. One is a smaller subunit, another one is a bigger subunit. Smaller subunit and a bigger subunit. Okay. So, simply you remember ribosomes, they were discovered by a scientist called Pallade. So, they are called Pallade particles. They mainly involves in the protein synthesis. So, they are called protein factories. Protein factories. There are two types of ribosomes. They are 70S and 80S type. 70S and 80S type. So, mainly you have to remember this one. 70S type of ribosomes are present in prokaryotes, mitochondria and chloroplast of the eukaryotic organisms. Whereas 80S type of ribosomes are present in eukaryotes. Another point you have to remember. So, I already told you the ribosomes are having two subunits. One is smaller subunit, another one is bigger subunit. Okay. So, you remember here 70S type of ribosomes, they are having the subunits called 30S and 50S. You remember this one, individually they are having 30 and 50. Okay. If they are condensed, they become 70S. Just you remember that one. Okay. Do not go in depth of that. 30S and 50S. Whereas 80S subunit, 80S ribosomes, they are having 40S and 60S type of subunits. This much is enough. So, just I will revise once. Ribosomes, these are called Pallade particles. They mainly involves in the protein synthesis process. So, they are called protein factories. There are two types of ribosomes, 70S and 80S. S represents the unit, Swedberg unit. Okay. So, the 70S type of ribosomes are present in prokaryotic organisms, that is in the bacteria, mitochondria and chloroplast of the eukaryotes. Then 80S type of ribosomes are present in eukaryotic organisms. So, I already told you the ribosomes are having two subunits, smaller one and a bigger one. And this 70S type of ribosome, they have two subunits, 30S and 50S, whereas 80S are having 40S and 60S type of subunits. Mainly you have to remember protein factories 70S and 80S, where they are present, 70S in prokaryotes, mitochondria and chloroplast, 80S in eukaryotes. This much is enough. Okay? Right. Next, lysosomes. Lysosomes. So, inside the cell there are small rounded structures. So, these are called lysosomes. Okay. So, membrane bound small structures are present. So, these are called lysosomes. So, these lysosomes contains many hydrolytic enzymes. They contains many hydrolytic enzymes or hydrolases, hydrolytic enzymes, or they are also called as hydrolases. Okay. Means they involves in the digestion process, you can say. These lysosomes, they involves in intracellular digestion. Intracellular digestion means if any foreign particle enters, into the cell, that will be engulfed, that will be destroyed by these lysosomes. Or if any cell organelle of the cell is damaged, that will be destroyed by these lysosomes with the help of these hydrolytic enzymes, with the help of these hydrolyzing enzymes. These hydrolytic enzymes means digestive enzymes. Is it clear? So, as they are also involving in the digestion of their own cell organelles, so they are also called as suicidal bags. Suicidal bags of the cell. Suicidal bags of the cell. So, simply you have to remember lysosomes contains many hydrolases or hydrolytic enzymes. They involves in the intracellular digestion process. They are also called as suicidal bags of the cell. Intracellular digestion means within the cell. Intra means within. So, if any foreign particle enters, that will be engulfed, that will be destroyed with the help of these hydrolytic enzymes. Okay, these are called lysosomes. Why they are called suicidal bags? Atmahuti sanchulu. Okay, suicidal bags ani antaru. So, because if any internal cell organelle is damaged, that will be cleaned up, that will be destroyed, further destroyed by these lysosomes. Is it clear? That is why they are called suicidal bags. So, that much is enough. Then vacuoles. Vacuoles. Suppose say this is a cell. Inside the cell, in plants, a large empty space is present. This is called as vacuole. Mostly present in the plants. Mostly present in plants. Mostly present in the plants. So, these are called dustbins of the cell. Dustbins of the cell. Why they are called dustbins? Because in plants various types of ergostic substances or excretory substances are released. Okay, waste products are released. So, as the plant does not have any separate excretory organ, so that cannot be sent out. So, they will be stored inside the vacuoles. So, that is why as these vacuoles are storing the waste products, that is why the vacuoles are called dustbins of the cell. Dustbins of the cell. Just you remember this terminology. Vacuoles are mostly present in the plants. In plants, a large vacuole is present. Okay, and this vacuole is surrounded by a membrane. That membrane is called a tonoplast. Tonoplast. So, you do not need this one, just to remember tonoplast. The membrane surrounding the vacuole is called as a tonoplast. The membrane surrounding the vacuole is called as a tonoplast. And these vacuoles are also called as dustbins of the cell. Dustbins of the cell. And another structure, centriole. Centriole present only in animals. Present in animals. Useful for cell division. So, near the nucleus, a small structure, a rounded structure is present, that is called as centriole. It is mostly present in animals. It is useful for cell division. So, these are the various types of cell organelles present inside the cell. Okay. So, just briefly I will revise once. So, if you observe the eukaryotic cell, it contains cell wall and protoplasm. The cell wall is present only in the plants, and protoplasm is divided into plasma membrane and cytoplasm. That plasma membrane is a thin membrane surrounding the cell. Okay. And inside the cell there is a fluid matrix that we call as cytoplasm, and inside the cytoplasm there are various types of subcellular structures are there. They are called as cell organelles, and these cell organelles they includes mitochondria, chloroplast, endoplasmic reticulum, Golgi complex, ribosomes, lysosomes, vacuoles, and also centriole. And the nucleus is there. Nucleus. That nucleus is the main cell organelle which controls all the activities of the cell.